But today we will discuss how to design our query or the target sequence by using homologous modeling and Swiss model. So first of all, open Swiss model. Uh, in Swiss model, the general principle behind the routine designing or the 3D structure modeling of target sequence is it first blast the target sequence in the PTV and the more appropriate similar sequencing used as the template and design the target sequence. First of all, open start model. Then paste our target or query sequence. So uh, let's consider this is our target sequence and paste in on it. This is our target sequence which was used to design the 3D model of that sequence. Uh, we can design the sequence by in two format. First, single target experiment. That means in that sequence used as one target or multiple target sequence. That means the sequence is clipped in two part, one and other. So first we will learn for single target. Uh, then project title. Let's consider this is the ornithine. Now let model. Then click on search for template, not directly click on build model. Because in build model, if we first click on build model, the Swiss model can design the 3D structure according to him. But if we click on the search template, uh, first the sequence is blast on the PDB sequence and many template or RHS. Out of several templates, we can choose which template is appropriate for our target sequence to designing this model. So it takes some times. For some time, this is our result. Uh, our the target sequence in Swiss model gives several templates, and we choose those templates whose GMQE value is nearly equal to 1 and identity. That means our target sequence is similar with the template sequence. How much? Then the method. Method is X-ray. X-ray means this structure is experimentally validated, but AlphaFold means it is computer-based design. So we choose the X-ray structure for our homologous modeling. First, click on it. And this is the template structure. According to this template structure, our target sequence model. Then click on the build model. Let go. You can choose that sequence or that this template for design structure, or you can also select the two sequence or the two template for designing the protein structure. Let single. Then click on build model. It takes some time. And after that, you can find your the 3D structure of the target sequence. Okay, and how you how you can select that sequence or that structure for for downstream experiment. Downstream experiment, uh, this structure is stable or not for docking purpose. That depends upon the GMQ value and local quality estimation. The local quality estimation, that means it can determine how much similar the model structure with the template structure. The x-axis represent the number of amino acid residue, whereas the y-axis represents the similarity. If the similarity is 1, this is the best model. You can find that all the sequence of the model structure is nearly equal to 1. That means this structure is best structure. And only the residue 210 to 220, this is the less similar. That means it is the disorder or the flexible. And also you can choose let if all the sequence it lies below the 0.5, then that structure not take for further docking purpose. Then we also choose the z-score value. That means if the star represent the model structure, if the star is present that region, we not take that structure for docking purpose or for the analysis purpose, because this that region represent a highly disallowed. That means the structure is not a properly folded or the properly designed or modeled for the analysis. Mm. And this is the sequence similarity. Then we can download this structure in PDF format. This is our model structure. So we can use homologous modeling in Swiss model, iTeaser, and modelers. Today we will learn how to model the target a query sequence of a protein into a 3D structure by using Swiss model. And in our next class, we'll learn how to model this sequence in modeler. After that, we'll learn in iTeaser. So thank you. If you have any doubt, you can comment or message me. Okay. Thank you.